Harvard lab before this, and he leads an effort to understand how the brain analyzes complex data sets streamed from the different organs, and he'll enlighten us today on the theory of neurocomputation. Off to you, Dmitry.
All right, so let's get started. I'm still trying to work out the system, but you know, if let me know if you can't hear me or something is not clear, and I'll try to respond. So I think it's fair to start with noting that the ability of our brains to make sense of the world and to come up with new ideas far exceeds the ability of even the most powerful modern computers. But how the brain is accomplishing all these feats is not clear. And I'm sure you've heard a lot about different experimental observation and biological facts about various aspects of the brain computing. And in fact, we have a lot of those. But I think what is sorely missing is a theoretical framework akin to the one we have in physics and other physical sciences that would allow us to put all the experimental data in one framework and make sense of the observations. And what I would like to do today is to present our progress towards such framework. And I would like to start with this quote that I think defines what we are trying to do in the best possible way. This is the quote that comes from perhaps one of the most famous physicists of the 20th century, Richard Feynman. In general, we look for a new law by the following process. First, we guess it, then we compute the consequences of the guess to see what would be implied if this law that we guessed is right. Then we compare the result of the computation to nature or experience, compared directly with observation to see if it works. If it disagrees with experiment, it is wrong. That is all there is to it. So this seems like a clear and simple prescription, and it worked amazingly well in physical sciences. But I'm sure if there are biologists in the audience, they're going to say, well, but just because this prescription works in physical sciences doesn't mean it's going to work in biology. And what I'm going to do today is to demonstrate that this same approach will actually allow us to make progress in biology. And we will actually use the same kind of laws that I used in physics, in the sense that these laws will be formulated as an optimization objective. So we will formulate an objective, and we will try to optimize that objective, and then hopefully we will come up with something that looks like biology. Of course, this is not physics, so the objective here will not be based necessarily on the principles of symmetry, but we'll have to do more with statistical learning and signal processing, things that we borrow from electrical engineering and computer science. So the talk will consist of several parts. So I will start with the representation and decoding framework, which has been well established in neuroscience and has a great record of success. And then we will switch to the framework which we call similarity matching, which is the approach that we've been developing recently, and the motivation for this switch will become during the talk. So historically, one of the first and most profound observations about neurons as statistical learning devices was the suggestions that neurons perform principal component analysis. This is obviously an oversimplification, but it's a good place to start. So I thought that this will be the first topic. I suspect that most of you know what principal component analysis is, but just to make sure we are on the same page, let's quickly review. So suppose you have a set of data points where each data point is a vector, for example, in a two-dimensional space, and the components are x1 and x2, and there isn't just a single data point, but there are several of them, a cloud of data points, and you would like to represent that cloud in an efficient manner by associating only a single scalar with each data point. Then what you can do is you can identify the principal direction that captures most of the variance in the data set, and then project each data point onto that direction. Then you can represent each data point by the length of the projection on this principal direction. Importantly, one can view this procedure as a result of solving an optimization problem shown on the right, where we minimize the sum squared error over all data points with respect to the principal direction W and the principal components YT. So for every data point XT, we try to find the optimal representation, which in this case is a vector W weighted by YT. What does this have to do with neurons? Well, the idea is that the input vectors XT are the activities of the upstream neurons from which a given neuron receives connections. So the coordinates x1, x2, and so on are, for example, firing rates of the upstream neurons, and the principal component is what the neuron computes. It's the output of a neuron, its firing rate, y. By the way, you probably heard about spikes in neurons. In this talk, we will ignore the spiking completely, not because it's not important, but because for our purposes, we can just think about firing rates, which is the number of spikes fired in a unit time. So what this model suggests is that a neuron computes the principal component of the upstream activity, y, and outputs that principal component. Of course, this view of PCA that I just described isn't quite appropriate for a neuron because this is a view of a statistician who has the whole data set in front of him when he starts running an algorithm. The neuron doesn't have such luxury. The data points for a neuron arrive one at a time, sequentially, and the neuron is required to output the principal component y before it sees the whole data set. Y before the next data point arrives. Then when the next data point arrives, you have to compute a new y, but also you have to adjust the principal direction W. And this kind of setting is called online. And that's the kind of setting that neurons have to operate because of biological demands. The other aspect of online computation is that, unlike statisticians who can store the whole data set in memory, the neurons don't have access to large amount of RAM, and they have to compute the corresponding principal component based on just the current values and perhaps a few state variables they could store biologically. So these are the additional constraints on the PCA that the neurons perform. And in fact, this kind of constraints have been already considered, and algorithms exist that are called online PCA. And those algorithms do the same thing that I just described. They optimize the same objective function, but they do so online as the data points are presented one after another. How to do this? One can solve this objective by performing two steps in response to each data point presentation. First, we can find, there is an animation that illustrates this process, but somehow it doesn't run. Can you see that? So with every data point presentation, you have to compute a projection y and adjust the principal direction w. And this can be done in two separate steps. The first one by minimizing with respect to y the squared error of representing only the current data point. This can be done in a closed form by a few simple algebraic operations, which if you don't follow, don't worry about it. I'm just putting it out there to show how simple it is. It just amounts to completing a square, and we find that the optimal yt, the principal component, is, of course, the projection of xt onto w, which is a normalized inner product between w and x. In other words, the output of a neuron is a weighted sum of the upstream neuron's activity. And now you start to recognize what you might have heard a neuron does. That is, sums up its input with the weights corresponding to its synapses. One point I should make is that in the objective function that we started, w multiplies the y, not the x. But when the online algorithm is derived, it turns out that w multiplies x, as you're used to produce y. So this is the first step of the algorithm where it computes the principal component. Now we have to adjust the principal direction to take into account the latest data point. And this is done by minimizing the cost function, which is again the squared error, but now summed over all the past data points. This again can be done algebraically, and you get a result that the weights of synapses are given by the covariances of the output neuron activity with the corresponding upstream neuron activity. This is known as a Hebb rule and has roots in biological observations. This is something that is well known in biology, that neurons that fire together wire together. But of course, when you look at this final expression, it doesn't seem to be particularly online, because it seems that the neuron needs to use all the past inputs and outputs to compute synaptic weights. But this is easy to take care of by algebraically transforming this expression into a recursion for synaptic weights, which looks like this and goes under the name of Euler rule in neuroscience, where you can see that the synaptic weight update involves this Hebbian term, which is the product of the postsynaptic neuron activity and presynaptic neuron activity, and the normalization term divided by what's called the learning rate, which is the cumulative sum of neuronal activity. This sum can be computed and held by a state variable of a neuron, so it doesn't require storing all the past data and can be just stored by a single state variable, which would be some calcium-dependent phosphorylation, for example. So what we did, we were able to derive so-called Hebbian learning rule for synapses that would implement the principal component analysis in a neuron. Now it's a good time to do a reality check and compare this with the experiment. As I already mentioned, the Hebbian term, which is the covariance of y and x, is rooted in biology, so that's totally plausible. But now we have another prediction, which Oya didn't have originally, about the learning rate. The fact that the learning reduces with time proportionally, inversely proportional to the cumulative activity of the neuron. And in fact, when experimentalists measure the ability of synapses to change their weight, they find that in long-term potentiation experiments, the synapses change less in response to the same stimulation in brain slices from older animals. And this dependence is at least consistent with the inverse proportionality with age. Of course, here we predict also dependence on the neuronal activity, and such experiments have been done as well, and it is known that if you suppress activity in developing animals, then the synapses stay plastic for longer, just as this theory would predict. So at least this kind of learning rule is consistent with what is observed experimentally. I don't want to overstate this agreement because there are many kinds of synapses and their learning rates depend on time and activity in different ways, but at least this theory has some legs and some ability to explain and understand this kind of observation. So to summarize, what we have done here is we started with the computational objective, such as a principal component analysis, which is based on infinite squared representation error of the linear representation in this case. We derive an online algorithm that optimizes this objective. And this online algorithm has two components. The first component is the neuronal activity dynamics, which is the weighted summation of the upstream neuronal inputs, which is, of course, what neurons do, where there are synapses, whose weights are the components of W. And then the components of W are updated, usually on slower timescales, based on the correlation or covariance in activity of pre- and postsynaptic neurons. And so we can map this online algorithm onto the neuronal computation in a single, oversimplified, but reasonably useful linear neuron. I don't know if it is possible for me to take questions, but this would be a good place to stop and ask for questions. The second term, WTYT, is there because of the normalization of synaptic weight, and it suggests that the update of synaptic weights depends also on the previous synaptic weight times the square of the postsynaptic neuronal activity. Now, this is something that is, in principle, biologically plausible because the neuron has access to both, the synapse has access to both its synaptic weight and the postsynaptic neuron activity. But whether or not this term is exactly what happens in neurons is not really clear. So again, I don't want to overstate here, just want to say that this rule is biologically plausible, but I don't think we can say there is a smoking gun saying that this is exactly the right way to look at synaptic plasticity. All right. So this theory that I described to you is based on Euler's proposal from more than 30 years ago, and it does give you a very oversimplified but a reasonable starting point to think about what neurons do. A very rather unsupervised algorithm that computes principal component of the upstream neuronal activity. But of course, we all know that you don't want just the first principal component. When you do data analysis, you would like to have several principal components. And fortunately, our brain has more than one neuron. So we could compute multiple principal components by invoking multiple neurons. And this, of course, is particularly interesting these days because experimentalists are now collecting activities, recording activities of large populations of neurons. And one particular technique that I want to mention is in vivo activity monitoring using calcium imaging. In this technique, you can use an animal that has been genetically engineered to express activity or calcium indicators in its neurons. And then, if you have that neuron view a computer screen with a stimulus and put its brain under the microscope, you can see the neurons literally lighten up. And this happens because calcium concentration of neurons reported by the indicator reflects the activity of engineered neurons. At this point, I would like to run this video, and if you can launch it on your end, I think it would be better. So what you're supposed to be seeing is the circles flashing, and each circle corresponds to an individual neuron in a visual cortex of a mouse that is viewing a screen on which a stimulus is presented. And the corresponding stimulus is shown in the upper right corner of the video. There are two messages that I would like to take you away from this video. One is that in response to different stimuli, different neurons become active. So each neuron has its favorite stimulus. And second, in response to most stimuli, only a small fraction of neurons is active. So the neuronal activity is sparse. So now we are ready to go back and formulate the so-called efficient representation principle, which suggests that when a brain is shown a stimulus, such as this image, the activity of its neurons perhaps encodes the stimulus in a way that the features of individual neurons weighed with corresponding neuronal activities reproduce the stimulus. So the feature of each neuron is just the stimulus that stimulates this neuron the most. In the visual cortex, such features are edges or Gabor patches and of different orientation. And in the image on the right, the preferred orientation for each neuron is color-coded. And then the features of each neuron are shown at the bottom, where a patch of the visual scene is represented as a weighted sum of the features of individual neurons. And they're weighted by the activity of neurons represented by their firing rates, Y1, Y2, Y3, and so on. So I'm not saying here that, the brain images the world for a homunculus to look at it and make sense of it, But linear decoding is a theoretical or a computational device to make sense of neuronal activity in the brain by saying that that neuronal activity represents the presented image as a linear combination of the feature vectors weighed by neuronal activity. So this is the same representation framework that we used for PCA, except that applied to the large population of neurons. We can put this in a mathematical form by using linear algebra, and then the input data, which is this patch of the image, can be reshaped into a vector x, where each component of the vector is the pixel intensity of the presented stimulus, and that vector is now represented as a weighted sum of feature vectors, each of which is a column of the so-called dictionary matrix W. Again, each column here is the reshaped set of pixel values in the feature patch, multiplied by the vector of activities. Is this clear? Okay, so then we can attempt to find the appropriate dictionary matrix W and the neuronal activities Y by minimizing the same objective function that we had before, except that generalized for a population of neurons. So now the output of the system is not just a single scalar per data point, yt, but it's a vector yt, which consists of the activities of the population of neurons, and it's multiplied not just by a feature vector w, which was the principal direction before. Now it's the combination of feature vectors, which is a matrix w, capital W. But the idea is the same, that we try to minimize the representation error by the ensemble of neurons. And again, we're assuming that the decoding is simply linear, so the representation of the stimulus xt is just a linear combination of the features with the weights corresponding to neuronal activity. And then what we have to do is minimize with respect to yt for each stimulus presentation xt, and minimize with respect to the dictionary matrix W for all the errors of representing all the stimuli that have been shown to the system. In the brain, synapses, each individual synapse cannot change its sign. It's either positive or negative. But so far in this formulation, in this objective function, the weights can actually be both positive and negative if you do not restrict the sign of x and y. So just like when you do PCA, you usually center the input data, and so x can be positive and negative, and so can y. And then w can change sign. Now, if you were to use x, which is non-negative, and y then will be non-negative, then w will be non-negative as well. And we will get to an example of that, which is much more biologically plausible. But at this point, you don't need to worry about it. One thing that you might have noticed here, which is different from PCA, is that the dimensionality, the number of the output components, exceeds the number of input components, which doesn't make sense in PCA because you don't want to exceed the dimensionality of the input space. But the reason I chose to do it this way is actually motivated by biology, where it's commonly observed that the number of neurons representing the stimulus exceeds the dimensionality of that stimulus. For example, the number of neurons in the visual cortex is more than 100 times greater than the number of input fibers that correspond to pixels, say, in the image. And so actually the representation is over complete. And this observation, coupled with the fact that neuronal activity is sparse, led Olshausen and Field about 20 years ago to formulate their famous theory, which is, I think, the pinnacle of the representation approach, where they suggested, as we did in PCA, that the brain minimizes the squared representation error, just like I showed before, but it does so using sparse neuronal activity, which is motivated biologically. And so the objective function they formulated combines the representation error term that we used in PCA with an additional term called a regularizer, which is just the L1 norm of the neuronal activity vectors, or simply the sum of neuronal activities. One can think of the reason to include this term is the need to minimize the energy consumption of the brain by reducing the fire rates of the neurons. And this term, interestingly, leads to a sparse representation using an overcomplete dictionary. And that's why W is called a dictionary rather than a basis, because it's overcomplete. Its components can't really be orthogonal. And so people use the word dictionary for it. So what Olshausen and Field did, they wrote down this objective function, which is supposed to produce a sparse overcomplete representation of the stimulus. But in addition, they were able to optimize it using an online algorithm. Now, the way to derive this online algorithm is rather similar to PCA, where first we minimize with respect to YT, that is the output corresponding to each input. And it can be found by iterating a gradient descent step, and you can recognize the terms in the brackets as the derivative of this first term of the objective function. And the reason you have the soft thresholding function shown on the right is because the second term of the objective is actually non-differentiable. It has a cusp at 0. And the proper way to deal with this in optimization theory using so-called subdifferentials leads to this nonlinear transformation on the derivative step of the first term. And so this might look like a hopelessly complicated expression, but actually it can be implemented naturally by a biologically plausible circuit of this form. So now, instead of having a single neuron that performs a weighted summation of upstream stimuli, we have a set of multiple neurons. Each weighs the input by this column of the dictionary W, and that's the first term, W transpose X. But then there are other inputs to the neurons that are produced by the outputs of the other neurons in the system. And that's the Gramian of W multiplied by Y. And then the sum is passed through a nonlinearity in a neuron, which can be modeled by soft threshold. So aside from the sign issue, this looks very biologically plausible. So biological neurons have a nonlinear function applied on the weighted sum of the inputs, but the activity, the output activity, cannot change sign. It is usually just positive. And so if you restrict, in addition to this, Y to be non-negative, then you get a rather biological looking network that corresponds roughly to something that happens in the visual cortex. Of course, computing the output activity is not the only thing that one needs to do in response to a single stimulus presentation, we should also update the dictionary W, which is encoded by the synaptic weights impinging on those neurons. And by taking a derivative of the objective function with respect to W, we can derive learning rule, which looks similar to the Oyer's rule, except that now we have this summation in the second term. So what Olshausen and Field did, they used this online algorithm where they alterated the neuronal activity dynamics, the first update, and then the synaptic update for each stimulus presentation on the data set of natural images, just like the one I showed before. So they took pictures of the forest, and they presented those pictures to the algorithm. And the big surprise came when they looked at the W's corresponding to individual neurons. So these squares are the features that each neuron learned in the algorithm. And those features are, surprise, surprise, Gabor patches of different orientation, spatial frequency, just like the features measured experimentally in cats and monkey brains. And so this algorithm was able to learn the features akin to the features represented by individual neurons in the actual brain without any supervision, just by being shown the natural images that our brains are exposed to in development and in evolution. So that was the big success of the Olshausen and Field theory, and that's why it made a big splash when it first appeared about 20 years ago. That starting with the principal objective function, like one would in physics, we could derive an algorithm that, when exposed to natural stimuli, would produce the feature vectors, the properties of the neurons, and the circuit that is biologically plausible or somewhat biologically plausible, and reproduces the observation in actual animals. But there was a problem with Olshausen and Field. And the problem has to do with this learning rule, and in particular with the second term, which Marta asked about, Euler rule, where it didn't have a sum. Now it has the sum, and if you look carefully, this sum is over all activities of all the neurons, not just the two that this synapse Wij connects. And so what this learning rule requires, that this poor little synapse, the mince of your brain, that connects only two neurons, updates its weights based on the activities of all the other neurons in the system, weighted in some synapse-specific way. And this is something that is totally biological implausible, because that synapse doesn't have access to the activities of all those neurons weighted in this particular way. And so this is too much to ask from a synapse, and is hopeless to implement biologically. And this biologically implausible feature of the Olshausen and Field model has been known, and in fact, it has been realized by Olshausen and Field, and that's why they actually proposed a different architecture to implement their online algorithm, but that architecture doesn't map well on the visual cortex. And so there have been this annoying problem with this beautiful theory that the learning rule has been non-local. There have been many other modifications of the theory, starting with slightly different objective functions, but none of them produce biologically plausible local learning rules. People, of course, were frustrated by that, and they instead tried to put in local learning rules heuristically without deriving them from an objective function. So they abandoned the objective function and then had local learning rules. You can have that, but then you kind of lose this theory. you lose this principled approach and the ability to analyze what the algorithm does. And so there has always been this disconnect that you could not have a neuronal network that performs either sparse representation or principal component analysis where the number of components is greater than one with local learning rules. And that was a problem that bothered me a lot. And so we've been thinking about this for a very long time. And eventually we realized that the way to overcome this problem and to have a principal objective function from which one can derive local learning rules would be to rethink the computational objective that the neurons are trying to achieve. So we went back to the beginning, right? Just like Feynman said, we have to guess the objective, right? And we guessed a different objective. And it turns out that from that different objective, we were able to derive the local learning rules. So what I'm going to do now is I'm going to tell you a little bit about this new computational objective that gives local learning rules. But I want to ask you about the time constraints first. How much time do we have left? So what is the similarity matching principle? It's actually very simple. If you have a network of neurons, we are postulating that similar inputs to that network will produce similar outputs. And the dissimilar inputs produce dissimilar outputs. So consider, for example, stimuli which are different breeds of cats and dogs. And of course, cat breeds are rather similar to each other, but dissimilar from dog breeds. And so we have the similarity matrix on the left, where the cat breeds correspond to this block of high similarity, and the cat to dog similarity is low. And what we're postulating is that this similarity must be represented by neural activity patterns. And if you only had two neurons, for example, then one of them would be a cat neuron, another would be a dog neuron, and then the neural activity patterns would be one on, the other one off, and then the similarity matrix would have the simple rank 2 form, which would approximate the input, okay? So that's the basic idea of similarity matching. Sounds like a simple principle, but doesn't have any biological justification. And it turns out, in fact, we didn't know that when we came up with this theory, but it has already been reported that the brain does similarity matching. And I'm going to show you this experimental results obtained from functional MRI imaging on the visual cortex in humans from the area called inferior temporal cortex. So the way this experiment was done is they presented 100 different visual stimuli to the human subjects in the MRI scanner, and they recorded the distribution of neuronal activity in response to each stimulus. Of course, the resolution of the fMRI scanner doesn't allow you to detect the activity of individual neurons. It coarse grains the activity over voxels, which have about 10 to the 5 neurons. But if you think about voxel activities as components of the activity vectors and then compute scalar products between the activity vectors corresponding to individual stimuli, thus compute in similarity, you can put all the results into this matrix where the color of the pixel reflects the similarity of activities in response to the corresponding pair of stimuli on the corresponding rows and columns. And of course, you can see that the activity is most similar between the stimulus and itself. So the highest similarity is on the diagonal, right? That's just the scalar product of the vector with itself. But in addition to that, you see this block diagonal structure where inanimate stimuli elicit similar activity to other inanimate stimuli, but different from animate stimuli. In addition, there is a finer structure corresponding to faces, both human and non-human objects. And so you can see that the similarity of neuronal activity is reported by fMRI actually matches the cognitive similarity that one expects between the stimuli. So there is a similarity matching happening in the brain. Now, this is, of course, this coarse-grained activity reported by fMRI, but similar experiments have been done by single electrode electrophysiology, and the similarity matrix looks almost indistinguishable at this level, and you can also see this log diagonal structure corresponding to cognitive stimuli if you record in monkey inferotemporal cortex. So there is experimental evidence that the brain matches similarity. Now, to develop a theory, we have to do a little bit of math. As before, we describe the input to the network as vectors, and what we require is that the similarity of those vectors is matched by the similarity of the output activity vectors. So vectors that are nearby in the input should end up nearby in the output. We describe similarity by the scalar product or the inner product between those vectors. Then those two should match between input and the output. Now, because this theory operates with the similarities between all possible pairs of inputs and outputs, it makes sense to use the matrix convention, where we have the data matrix consisting of the column vectors corresponding to each stimulus. And then using this matrix convention, we can write down the similarity matrix of the inputs as the Gramian of the data matrix, X transpose X. And the same for the outputs. Now note that this Gramian in our notation is different from the sample covariance. Sample covariance in our notation would be X transpose. This instead is the Gramian or the kernel matrix, X transpose X. And what the similarity principle says is X transpose X should be matched by Y transpose Y. They should be, the similarity matrices should be similar. Those of you familiar with multidimensional scaling may recognize that this is similar to the requirement of the classical multidimensional scaling, which is formulated by using this kind of objective function, which is the same idea as we had before. It's a sum of the squared error, but the error now is the mismatch between similarity matrices rather than the representation error. So that's the difference, okay? And so this is now our new objective function that we are going to use to derive the online algorithm that will map neurons. Of course, if this, the whole optimization problem, it has a trivial solution where y equals to x, and the way to avoid it is to regularize the problem by introducing the constraints. One way of doing that is to require that the dimensionality of y is less than x. So there are actually fewer output channels than input channels, as you would do in PCA, and that in fact can be solved offline and leads to linear dimensionality reduction, and y just projects the input data, the principal subspace, which up to rotation is PCA. So then you might ask, what did we do, right? The evidence that the brain does similarity matching existed from fMRI before, and the objective function can be taken from multidimensional scaling. Well, so what we did, we derived an online algorithm to solve this objective function that comes from multidimensional scaling and map it onto neurons. And because we are running out of time, I'm not going to go through the details, but we are using the same kind of approach, following the same steps that we used for single neuron PCA and for Olshausen and field theory, starting with the computational objective, which is linear dimensionality reduction. We write the objective function. We minimize it online by taking derivative, setting it to 0. And then what we find is that it actually can be solved by this iterative algorithm where y is updated based on the linear combination of the inputs and the outputs, just like before. And that can be done by a neuronal network, which is exactly the same architecture that Olshausen and Field use, having both Hebbian feed-forward and anti-Hebbian lateral connections. But the weights of those connections are updated by purely local rules. If you remember, in the Olshausen and field theory, we had this annoying sum over activity of all the other neurons in the synaptic weight update. Now, we don't have this sum. Instead, the each synaptic weight is updated based only on its own weight and the activities of the two neurons, xj and yi, that it connects. And this can be derived, again, from principal objective function, not quite by gradient descent, by the Newton-like method, but anyway, in a principled way. And so we were able to obtain local, biologically plausible learning rules from a principal objective function once we use a similarity matching as our objective. And this way, we were able to both have the cake and eat it too. So the algorithm that we derive can be run by a neuronal network like this with alternate activity iterations and synaptic weight up, and in fact finds the principal subspace no worse, but actually better than the existing neural network algorithms. So I should probably summarize here. The part that I'm going to skip is another way to regularize the objective function by requiring that the outputs are non-negative, which is, again, biologically inspired because the neuronal activity of iron rates, they cannot be negative. And the amazing fact is that if we impose this constraint, then we can derive the similar looking algorithm which is implemented by the network of the same architecture, but now that network performs clustering. Instead of PCA, it does clustering or sparse feature extraction just by introducing this constraint, and this is similar to symmetric non-negative matrix factorization, but done online. And so now we can reproduce the same results that Olshausen and Field have, which is an unsupervised learning of neuronal feature vectors from natural images by running a network which uses only biologically plausible local learning rules. Now, to put it all together, I want to say a word what we mean by similarity matching, because first, remember when I gave an example of similarity, I talked about high-level features and stimuli like cats and dogs, or cognitive representation. But now I'm talking about the similarity on the single pixel level, which gives you these Gabor patches. So which one is it? Well, the answer to this question depends on which part of the brain you look at. And the idea is that when the images come into the eye, they first are processed by matching similarity on the pixel level. And that's where we get Gabor features, just like you see in the visual cortex. But because the transformation involves nonlinearity, this rectification in neurons, for non-negative objective function, we actually transform the data into the new space, and then we can apply a similar kind of objective function to the outputs of the first layer in the second layer. That layer may learn higher level features. Once this process is repeated a few times, we may get to the higher level features, like the features of cats and dogs, like the length of their fur and the number of eyes and the length of the nose, the ratio of the length of a tail to the length of the legs, and so on. And so, of course, this is not something we have done, but this is work in progress, where we try to stack up the layers, each of which learns features on the corresponding level to arrive in an unsupervised fashion to higher level representations of cats and dogs, which will hopefully parallel what happens in the visual processing stream in the real brain. So to summarize the talk, I hope I was able to demonstrate to you that the same approach, eloquently articulated by Feynman, has been used in physics so successfully, can in fact be used in neuroscience. And by starting, or guessing rather, principal objective functions, deriving online algorithms, and comparing them with biological experimental results, we can make progress to understand and hopefully understand how neurons perform computation on the algorithmic level, but in close contact with their biophysics. And finally, I want to acknowledge the people who were central to this work. Brilliant postdoc Chengiz Pelivan, who has been driving most of this work, which has also been built on the work that has been done by another brilliant postdoc, Tao Hu, who has since left my group and moved on to an independent position. And I also have moved to the Simon Center for Data Analysis in New York City, and we are hiring. Thank you. Thank you very much.